So if you remember the very end of when we met on Tuesday,
Mary.
Yeah? Tell me you can hear me. No?
I can hear you.
Okay, good. So the very end of the meeting, or very beginning of the meeting, I said that I wasn't teaching 122 next semester. Well, there was a miscommunication, and I am.
Yay.
That makes me happy. But when I'm teaching it, it's on a Friday. And I need to look and make sure that I'm correct about the times. But it is from, where did I put it? So it's from 8 o'clock in the morning on Friday. It's like an all-day class.
Oh, 8 o'clock.
No, tell me about it. But you know what? I can make it if you can make it. I promise.
True, true.
Because I'm not, I can tell you that of all times, I am not a really great morning person. But it starts at 8 in the morning. The lecture is from 8 in the morning until 10:40 in the morning. And then the lab is from 12 until 2:40 in the afternoon.
Are you going to be teaching the lab as well?
I am. I'm teaching.
Please, which day is it, Ma?
It's Friday. So it's essentially it's a Friday all-day class because it's going to go from about 8 in the morning until 3 in the afternoon.
What's the name, please?
It's Biology 122. I don't know the CRN on it. I only figured it out the other day. Was talking with the bookstore and come to find out because he knew that I was teaching 122 and I didn't. And I rechecked with the chair of HAC and he relooked and yes, I am teaching 122. So it's on Friday from 8 a.m. till 10:40 a.m. And then the lab is that on Fridays from 12 to 2:40.
Okay, because the one you told us the last time, it's the time is 6 to 8:40 p.m.
That's a 121 course.
No, that's 122.
Are you sure?
Yes. Nancy.
Do you want to share your screen and tell me?
Oh my God.
Because I just registered for the Negli Root and Nancy Konoka.
Well, let me show you. I'll show you the email that I received from the chair telling me which ones I was teaching. Bear with me just for a second here. Okay. So looking here. And I'll tell you, I'll warn you ahead of time, it may be a very interesting semester because I have never taught biology 122. And so the first semester always teaching, of course, is always a little rougher than the rest of the time. But so this is the email that I got. Where is it? So
Yes,
bear
that
with me.
was the one I picked.
Bear with, I'm sorry, Toya, bear with me just for a second longer. I need because he told me which ones they were. Where is it? I thought he said.
The number is 38266 for registration purposes.
Because they're not even teaching. It's so annoying. Like they're talking about classes for next semester, like what she's teaching.
Hey, and I can hear what you're saying. I know it's annoying. That was funny. That I can tell you after class. I'm just telling you that's what I know so far.
Thank you for sharing that. I
I'm
appreciate it.
sorry, Professor. The one I picked is the CRN is 36853.
You know what,
For the-
Toyin? I don't know for sure. If I remember right, his email the other day told me that it was the Friday class. And
Yeah,
and
but
I may be wrong.
But the
But
one I picked, it was the same name you told us, Negli Roots and the
I thought
Nancy.
that was 121, but I may be wrong. I don't.
No, I found
but-
the name in 122.
Okay. If that's it, then that's it. I need to verify with him, all right?
Okay, because I found 6 to 8:40 p.m. on Thursday.
Okay.
But the lecture one is virtual.
Okay. Then you're probably right. It's a Monday, Wednesday course. I'll verify with him because I need to know what's what for sure. But let's move on into the lecture material, okay? I think we were about right here. So central nervous system includes the brain and the spinal cord, and they're enclosed and protected by the meninges, the cranium, and the vertebral column. The peripheral nervous system is all of the rest. So this includes the spinal nerves and the cranial nerves. So the peripheral nervous system is specifically bundles of nerve fibers wrapped up in fibrous connective tissue and ganglia, which are the knot-like swellings along the nerve where the cell bodies are contained. The peripheral nervous system includes both the sensory division and a motor division. The sensory division is the visceral sensory division. Remember, visceral meaning organs. So this is specifically sensory around the organs. Somatic includes skin, muscle, bones, and joints. So we have sensory for both the organs and then the somatic, and we have motor for both the organs and the somatic. One way to think of it is the visceral is the autonomic, and the somatic is the voluntary. Autonomic meaning involuntary. The sensory divisions, two areas right here, are going to carry signals from different receptors to the central nervous system, informing the central nervous system of changes within and around the body. So visceral for coming from sensory information from the organs, somatic meaning sensory information from the skin, muscles, bones, and joints.
You said somatic is involuntary, correct?
Somatic is voluntary,
Okay.
so the opposite. So visceral meaning tends to be all involuntary.
Okay. Thank you.
That's it. That's when we say it like that, realize that there are exceptions, but overall that's how it is.
Okay.
The motor or efferent division carries signals from the central nervous system to glands and muscle cells. The effectors are the organs and cells that carry out these responses. So the central nervous system sends a signal to the gland or motor cells that are effectors saying, this is what we want you to do. So the somatic motor neurons are going to carry signals to skeletal muscles. The visceral motor division is going to carry signals to glands, cardiac muscle, and smooth muscle. These responses that are carried out by the visceral motor system are what we call visceral reflexes. The two divisions to the visceral motor division, or also called the autonomic nervous system, are the sympathetic division and the parasympathetic division. The sympathetic division is your fight or flight response. It's where you get an accelerated heartbeat, increased respiratory airflow, eye dilation. But there's an inhibition in digestion. The parasympathetic division has a calming effect. This is the slowing of the heartbeat, stimulation of digestion, stimulation of the urinary system. Some fundamental properties about neurons. Neurons are excitable. They respond to changes in the environment. So they always are responding to different stimuli. They're conductive. They respond to stimuli by producing electrical signals that are quickly conducted to other cells. Secretion: when the signal reaches the end of the nerve fiber, the neuron reaches a neurotransmitter, secretes a neurotransmitter that stimulates the next cell. So they're excitable, they can conduct an electrical signal, and they can secrete neurotransmitter. So there's two main types of nerve cells. All together, we have neurons and we have neuroglia or glial cells. All together in the nervous system, there in every individual, there are about 1 trillion neurons. So just to put this in context, this is about 10, there's about 10 more neurons in Caitlin's nervous system than there are the number of the stars in the galaxy. The neurons, these 1 million neurons in one person, make up about 50% of the volume of the nervous system. These specialized cells transmit signals throughout the body. Glial cells or neuroglia play a major role in supporting and nutrition of the brain, but they don't manipulate the information. So the neurons are the only ones manipulating the information. These play a supporting role. These cells maintain the internal environment so that the neurons can do their job. Well, they do a lot more than that, but generally this is what they do. There are about 10 to 50 times more glial cells than there are neurons. So altogether, 1 trillion neurons in the nervous system. And if there's 10 to 15 times more neuroglial cells than there are neurons, then we're looking at there being 10 to 50 trillion glial cells. Don't know what you think of that, but I think that's a lot. There's different functional classes of neurons. We have sensory or afferent neurons or approaching neurons. These are specialized to detect stimuli such as heat, light, pressure, chemicals, and transmit information about them to the central nervous system. I'm going to stop just for a second and introduce a few terms here. Afferent. Remember, afferent means in this case, it always in the nervous system, it means sensory. So incoming information into the central nervous system. So incoming information or entering. Efferent, it's motor, and it means exiting or outgoing. So an afferent sensory neuron is an approaching neuron that specializes and detects information in the peripheral nervous system and transmits that information to the central nervous system. Each of these neurons begins in almost every one of your organs and ends at the central nervous system. Some of these receptors are sensory neurons themselves, and others will communicate with a sensory neuron. Interneurons are the neurons in the central nervous system. They make up about 90% of the neurons, and they lie entirely in the central nervous system. So afferent neurons and efferent neurons are both are found in the peripheral nervous system. Interneurons are found solely in the central nervous system. Interneurons receive signals from many afferent neurons, and then they carry out integrative functions and make decisions about how the body will respond to stimuli. And then they send a signal out via the motor neuron or the efferent neuron to the effectors saying, this is how you should respond. So when we're looking at this, do you see the different parts of the feedback loop? So we have an effector. Sorry, we have a sensor. We have an incoming signal. We have an integration center. We have an outgoing signal. And we have an effector. Every time. While studying for an exam, you reach for a beverage. To extend your arm, your what nervous system is activated?
Is it B as in boy?
I think you're right. Why do you say that?
Because you're actually telling yourself, your brain's telling your arm to move to grab the drink.
Right. And that's the so wait, I'm sorry, what did you say? Say it one more time. B as in boy or D as in delta?
I thought it was B as in boy.
Okay,
Or is it
that's involuntary.
no D? It would be D because
D
that's
is-
voluntary.
It's D because D is your voluntary.
Yeah.
Autonomic, the two branches of the autonomic, which is also your visceral,
Okay. I see
is
now. Okay.
parasympathetic and sympathetic. An example of a neural feedback loop. So sensory function, you can smell coffee. It stimulates sensory receptors in the nose. There's an integrative function. The olfactory information is relayed into the brain where it's processed, and then there's a decision, ooh, that smells good, we should maybe drink some. Motor function, the brain stimulates the contraction of specific muscles to allow you to grab the cup and drink it. Questions? Structure of the neuron. So they're looking at the basic structure of a neuron. There's a zoma or a neurosoma. This is a cell body or the control cell, center of the cell. It contains the nucleolus. If you remember, the nucleolus is what contains the rRNA and the ribosomal proteins and other organelles, including a rough ER that contains chromatophilic substances, so color absorbing substances. or Nisi bodies, and a cytoskeleton, which includes neurofilaments. There are no centrioles here, so neurons cannot undergo mitosis to replace lost neurons. So do you remember what the function of the centrioles were? I know that was a long time ago. The function of the centrioles were to put out mitotic spindles and be able to separate the chromosomes during division, during mitosis. So if there's no centrioles in the neurons, they're not going to undergo division. In addition to the organelles and the soma cell body, there's dendrites that branch off from the soma. I like to think of these as being little feelers. They just they extend out like little feelers, and they're finger-like projections that are branching off from the cell body. They're primary sites for receiving signals from other neurons. The more dendrites a neuron has, the more information it can receive and incorporate into making a decision about an action. One thing I'll also tell you is that the more stimulated a neuron is, the more dendrites it can develop. And so this is a very dynamic process by which, if a neuron is stimulated more, it will develop more abilities to receive information. Moving down towards the axon, there's an axon hillock, which is where the axon is going to originate from the soma. The axons are relatively unbranched, but there may be axon collaterals that branch off occasionally. Axons are specialized for rapid conduction of nerve signals. The cytoplasm is called axoplasm, and the membrane is called an axolemma. Axo means axon. Plasm means cytoplasm. And lemma means plasma membrane or cell membrane. So the axolemma is the plasma membrane of the axon. The axoplasm is the cytoplasm of the axon. The axon may or may not be myelinated. At the distal end of an axon, there is usually a terminal arborization. What does arbor mean? What does arbor mean? Where have you heard that term before? ARBOR.
Tree.
It means a tree. So terminal arborization is essentially a branch-like, tree-like formation at the end of the axon. So each of these branches is going to end in an axon terminal. Each of these little branches are going to end in an axon terminal that will synapse with another cell. I like to think of a synapse as a snap on a shirt. Two things coming together, one cell coming together with another cell. So each axon terminal is going to synapse with another cell. And in each terminal, there are synaptic vesicles that contain neurotransmitter. So this terminal arborization contains this extensive complex of fine branches where each one is going to terminate in a synaptic knob where it will form a junction or synapse with the next cell. And contains synaptic vesicles full of neurotransmitter. So each branch here. The vesicles of neurotransmitter can contain neurotransmitter that can be excitatory or inhibitory to the next cell. Can either inhibit the next signal or initiate it. Questions?
I have one just to make
Sure.
sure I'm understanding.
Okay.
So nerves, obviously, like, okay, you touch something, it's hot, it burns you, you pull away. So would that structure of that neuron be like not non-existent, but it doesn't have the centrioles to like reboot it? So is it just kind of,
You know what? Wait for that one. That's
Okay.
coming. That's coming.
okay,
Ask me that question like either today or next week.
okay.
That's coming because we will talk about regeneration a little
Yeah,
bit.
I
Depends
just want
on
to make
where
sure
it
that
is.
I'm like understanding.
Yeah, it depends on where it is.
Okay.
You can affect, you can see here, you can activate a nerve without destroying it also.
Okay.
So just because you got burnt doesn't mean that you destroyed the nerve.
Gotcha.
But there is, yeah, wait on that one, Caitlin, that's coming.
Okay.
Multipolar nerves have one axon and multiple dendrites. These are the most common type of neuron and consist of most of the neurons in the brain and the spinal cord. Bipolar neurons have one axon and one dendrite. They're only found in specific places, specifically your special senses, so your nose, your retina, and your ears. The retina is part of the eye. An anaxonic neuron has multiple dendrites but no axon. It communicates locally through their dendrites but produces no action potentials. So this is mainly an integration center. If you think about it, if it's not producing any signal, action potentials are signals going out from the neuron. These are mainly integration centers where they're going to collect information and pass it along, but they're not going to pass it along through long firing signals. These are found in the brain, the retina, and the adrenal medulla. One more class of neuron structure are unipolar neurons that have a single process leading from the soma. They carry signals to the spinal cord or senses. There's a one process that's a peripheral fiber, and one process that's a central fiber. The peripheral fiber, if you think peripheral nervous system, has a fiber that ends far from the soma, the cell body. And the signal travels along the peripheral fiber to the central fiber and then to the spinal cord. These are specifically your sensory neurons. Sensory neurons entering the spinal cord. In additionally, we have axonal transport in neurons, which is a two-way passage of proteins, organelles, and other materials along the axons using microtubule monorails. When the motor engine functions along these rails, so remember, microtubules are part of the cytoskeleton. So these are essentially roads throughout cells. They move in two different directions, just like the roads on the highway. Anterior grade movement is going to move from the soma towards the axon terminal. And the car or the motor engine that moves anterior grade is going to be kinesin. When things move from the axon terminal towards the soma, that's retrograde transport. And then the motor engine involved there is dynein. There are two speeds of axonal transport. The first one is fast, the second one is slow. Fast axonal transport occurs at about 200 to 400 millimeters per day. This two types of axonal transport are fast anterior grade. Remember, that's moving things from the soma towards the axon terminal. And fast retrograde transport, which is moving things from the axon terminal back to the soma. And fast anterior grade is going to allow the soma to provide materials needed at the axon terminal, like synaptic vesicles, like enzymes that are needed to break down extra neurotransmitter. Fast retrograde is going to allow things at the axon terminal to be moved back towards the soma, maybe to degrade things that need to be broken down. and informs the cell body of what are the conditions of the axon terminal. We have pathogens like the tetanus toxin, rabies, and the polio virus that take advantage of this fast retrograde transport. When things can be taken up by the sensory neuron and passed back to make you really sick. Slow transport only occurs in an anterograde process. And it's a stop and go process. It's like taking a direct flight versus a flight that has multiple layovers. So your fast is a direct flight. Your slow axonal transport is multiple layovers. So the plane may go as fast, but then you're stuck at multiple stops. And the speed along slow axonal transport is about 0.2 to 0.5 millimeters per day. Questions? Neuroglia are not electrically excitable. So these glial cells function to serve and protect and support neurons. Oligodendrocytes are the first type of neuroglia in the central nervous system. They have a bulbous body and up to 15 arms. I look at this little guy. This is him, the oligodendrocyte right here. I think it looks kind of like a spider with multiple extensions coming out, multiple arms. Each arm reaches a nerve axon and spirals around it. This is the myelin in the central nervous system. So each arm can form one myelin segment around one part of an axon. So up to 15 different extensions. So one oligodendrocyte can be involved in the helping of myelination of 15 different axon segments. This is important to understand for later. Three different parts, three different arms may be involved in axon, in myelinating the same segments of one axon, different segments of one axon. Or each arm may be involved in myelinating a segment on 1 of 15 different axons. Ependymal cells look like cuboidal epithelium. But they have no basement membrane. So if you've talked about tissues in lab, all epithelium has a basement membrane. We don't have one here with ependymal cells. So they look like cuboidal epithelium, but there's no basement membrane. They produce the cerebrospinal fluid that bathes the central nervous system and fills the cavities. They also have patches of cilia that help the cerebrospinal fluid to circulate. So these are involved in secreting, so oligodendrocytes involved in the myelin sheath. Ependymal cells secrete cerebrospinal fluid. Microglia are small macrophages. What is a phage? Let me back up. What's phagocytosis?
It's when like a cell eats another.
Did you say? Oh,
Yep, it's when a cell eats another cell.
Yeah.
So a macrophage is a big cell that eats other cells. So a microglia are big cells that eat other cells. They develop from monocytes in the immune system. Monocytes are a type of white blood cell. They wander throughout the central nervous system, putting out finger-like projections to look for cellular debris or other problems. And they phagocytize that dead tissue, microorganisms, and other foreign material. So these are the immune cells of the central nervous system. They tend to be concentrated in regions where there's infection, trauma, or a stroke occurring. They aid in repair by synaptic remodeling and changing connections between neurons. So in the central nervous system, we have oligodendrocytes, which are involved in the myelin sheath. We have ependymal cells that secrete cerebrospinal fluid. We have microglia that are the macrophages that perform phagocytosis in the nervous system. And we have astrocytes. Astrocytes, from their name alone, astro meaning star-like shaped. They are many branch star-like cells. Constitute over 90% of the cells in some areas of the brain and cover an entire surface of the brain and most synaptic regions. Most non-synaptic regions. These are the neuroglia in the central nervous system that have the most number of functions. And we're learning more and more that they have even more functions than what we know now. So this is just 9 functions that they have. And we're learning that they have even more functions than this. Lots of different functions. So astrocytes provide the supportive framework for neurons. They have perivascular, peri meaning around, vascular meaning veins, capillaries. So they have perivascular feet that contact the capillaries and stimulate the formation of the blood-brain barrier. They monitor neuronal activity and stimulate dilation and constriction of blood vessels to regulate blood flow, to adjust to changing needs. So they're involved in regulating dilation and constriction of blood vessels to maintain changing needs. They also are involved in converting blood glucose to lactate. The brain can only live on glucose as a sugar source. However, the neurons cannot incorporate glucose. It has to be converted to lactate or lactic acid, which this occurs by the astrocytes. So the astrocytes metabolize glucose to lactic acid to allow the neurons to take up the lactic acid for nutrients. Astrocytes also secrete nerve growth factors that regulate nerve development. They promote synapse formation and fine-tune circuitry. They communicate electrically with neurons and influence synaptic signaling. They regulate tissue fluid so they can absorb, release neurotransmitters and potassium to prevent either of these becoming too excessive in their amounts. They also the last function is that they're also involved in performing sclerosis or astrocytosis. So filling space that was formerly occupied by damaged neurons with heart scar tissue. If you think multiple sclerosis, if you're familiar with that, those are plaques of scar tissue that have been created by astrocytes. Which of the following is not a function of astrocytes?
Would it be C?
Why? Why do you say that, Kevin?
Does the microglia do that?
Yep, you're
Or-
right. That's what the microglia do. Correct.
Whew.
In the peripheral nervous system, so in the central nervous system, the 4 different neuroglia, oligodendrocytes, ependymal cells, astrocytes, and microglia. In the peripheral nervous system, there's only 2 types. There's Schwann cells and satellite cells. Schwann cells or neurolemmocytes wrap repeatedly around the nerve fibers of the peripheral nervous system, producing the myelin sheath. These are analogous to the oligodendrocytes in the central nervous system. They're both involved in creating the myelin sheath. Schwann cells do it in the peripheral nervous system. Oligodendrocytes do it in the central nervous system. They do it by a little different manner, but they're the same. But this is their analogous functions. Satellite cells surround the cell bodies in the ganglia of the peripheral nervous system. They provide insulation around the soma and regulate chemical environments of the neurons. Questions about neuroglia? Gliomas, aka brain tumors, are almost always caused by glial cells. Why? Why are they almost always caused by glial cells and not neurons?
Because neurons rarely undergo mitosis.
Yeah, I mean, if the cell isn't going to divide, how is it going to go into uncontrollable growth? Also, they tend to grow very rapidly and become highly malignant. They're also tend to be resistant to chemotherapy because of the blood-brain barrier. Remember, most of the time when people receive chemotherapy, they're receiving it via the bloodstream. Well, because of the blood-brain barrier, it's hard to fight gliomas with chemotherapy. This picture right here, this is actually my cousin. And he had a brain tumor removed almost about 2 years ago. Was it 2 years ago? It may have been a year ago. It was about a year ago. And he's actually doing really well at this point. He just ran the St. George, Utah Marathon just about 2 or 3 weeks ago. But so he's doing really well at this point. But he's probably about 53, somewhere in there, not very old. So the myelin sheath, this is just like the insulation around the electrical wiring in your home. Why do you have insulation around the electrical wiring of your home?
So the wires don't cross and it doesn't catch on fire.
Well, yeah, hopefully it won't catch on fire. Why
A protection barrier.
So
is
you
it
don't get shocked.
So you don't get shocked, and it's protection barrier. What is it protecting you against?
The electrical current.
Yeah, the ions. Go get her. She's coming on the door. Go get her. So the myelin sheath is essentially the same thing. It's a layer of insulation around the nerve fibers. Think nerve fibers are axons formed by oligodendrocytes in the central nervous system and Schwann cells in the peripheral nervous system. The composition is about 20% protein and 80% lipid. Let me just for a second here. So I have an axon here. Pretend that my pencil's an axon here. Right. And I have a really, really long cell. That's my tape here. And I keep wrapping around it really, really tightly. Keep wrapping this cell around it really, really tightly. Really, really tightly. And I can wrap it up to 100 layers around this. Now, if this is a cell just membrane? Is a cell just membrane? Someone tell me, when you think about a cell, which we talked about cells, Are they only membrane? What else are they?
You have a nucleus, cytoplasm.
Right. So they have a nucleus, they have a cytoplasm. Well, if I wrap this around this membrane around this, wrap this cell around this axon really, really tightly, where's all the cytoplasm? Where's the nucleus? It gets squished out to the outside.
Oh.
So the insulation that is the myelin is essentially the plasma membrane. So it's a lot of lipid. Remember, the plasma membrane is made of phospholipids, so it's a lot of lipid. The production of this myelin sheath and the wrapping up around of the myelin is called myelination. It begins in about week 14 of embryonic development. But at birth, there's very little myelin in the brain. However, it proceeds really rapidly during infancy and isn't completed until late adolescence. Myelin is very much involved in synaptic formations and making connections between different neurons and maintaining those connections. Because of the need for this myelination that's occurring really rapidly, dietary fat is really important to early nervous system development. This is why children under the age of 2 it's not advised to give them anything but whole milk for this reason. Questions? So the Schwann cells, like my piece of tape there, can lay down up to 100 compact layers of the plasma membrane. The most outer layer is the neural lemma. Remember how lemma meant plasma membrane or the outer membrane? So the neural lemma meaning the most outer membrane. This contains the nucleus and most of the cytoplasm because everything else has been squished to the very most outer layer. Surrounding the neural lemma is a basal lamina and a fibrous coat called the endoneurium. So this is Schwann cells. With regards to the Schwann cells, it's one cell, one segment. One cell, one segment. In the oligodendrocytes, one oligodendrocyte, remember it can put out to like 15 arms. So one oligodendrocyte can be part of myelinating several nerve fibers. Since one oligodendrocyte myelinates multiple fibers, unlike the Schwann cells, they're anchored in place. So they can't move from one area to another area because they're connected to multiple axons. The Schwann cells, one cell, one segment. Oligodendrocytes, one cell can do up to 15 segments on different axons. Another thing that we see here with oligodendrocytes is they push new layers underneath older layers rather than having newer layers being formed on top of older layers of myelin. You get newer layers being pushed underneath older layers. So the myelination, rather than being formed from the inside out, is being formed from the outside in. So in the central nervous system, there is no neurolemma or endoneurium. Remember, the neurolemma is the most outer layer. Keep this in mind for a moment here. Many Schwann cells or oligodendrocytes are needed to cover one nerve fiber. Since the fiber is much longer than the reach of a single glial cell. The myelin sheath is segmented. There are gaps called nodes of Ranvier that are separated into internodal segments. So because it's not continuous, there's gaps. This is going to be really important next week. The short segment between the axon helix and the initial segment of the first glial cell is called the trigger zone. And this is important. It's not myelinated, and it's important in initiating a nerve signal. One thing to talk about really quick, because we're talking about myelin, is multiple sclerosis. This is an autoimmune disorder that attacks the myelin. Autoimmune meaning self-attacking, when your immune system turns on yourself. When it no longer, when it considers your self proteins to be foreign and therefore a danger to the body. So this is specifically when your immune system is attacking your own myelin. It can be triggered by a virus in genetic, it may be triggered by a virus in genetically susceptible individuals. And the result is that the myelin sheaths deteriorate in the central nervous system. And as they're broken down, they're replaced by scar tissue, which results in sclerotic plaques. Remember, that's the astrocytes creating the scar tissue. The result is that there's a disruption. It's just kind of like having your insulation, rubber insulation torn away from electrical wires. All of a sudden, there's shorts in the wires. They're not moving as fast as they should. Most people die within a year. Excuse me, few people die within a year of diagnosis, but others can live for 20 to 30 years. Now I want to hit on almost one more thing before we're over today. Each neuron in our body has a span of how long?
D.
Isn't it D?
It's D because they don't really divide, so they have to be that way. Maybe we won't get to what I wanted to. Unmyelinated nerve fibers. So we have nerve fibers that are not myelinated. Many nerve fibers in the central nervous system and peripheral nervous system are not myelinated. Well, how are they organized? In the peripheral nervous system, unmyelinated nerve fibers are enveloped in the Schwann cells that are wrapping myelinated fibers. So little folds in the neural lemma of the Schwann cells that are wrapping another axon can enclose very lightly an unmyelinated axon. So the neural lemma of the Schwann cells that are wrapping around one axon can envelope an unmyelinated axon. So one Schwann cell that's wrapping around one axon can harbor up to 1 to 12 unmyelinated axons within its grooves. Some terms to remember. Neuronal cell bodies are often grouped together in clusters. A ganglion is a cluster of cell bodies in the peripheral nervous system. A nucleus is a cluster of neuronal cell bodies in the central nervous system. In the peripheral nervous system, a bundle of axons is a nerve. A bundle of axons in the central nervous system is a tract. Nervous tissue is grouped into gray matter and white matter. Gray matter includes cell bodies, dendrites and unmyelinated axons and neuroglia, and it lacks myelin. The white matter is the myelinated. The lipid part of the myelin gives it the white appearance. The white matter is formed from aggregations of myelinated axons. So everything that's white matter is myelinated axons. Gray matter is everything else. Let's end with this question. Nerves are found in the what, while tracts are found in the what?
Is it A?
It's A.
A.
Let's end there. Any questions? I will see you guys next week. And I'll send out an email.
Thank you.
Yeah, let me find out for sure just what time that class is, because Toyin's probably right, but that's not what I was told in the email. So I need to make sure, and then I'll let you know. Okay, have a good day.
I wanted to remind you, Tuesday, I'm gonna try to make
You're going
class.
to Las Vegas.
Yes.
Okay.
So depending on, we leave at 6 a.m. from Baltimore, and we have a layover. So, and I know, like, with the time change and stuff, depending on if we're in the airport or on the plane and with like Wi-Fi connection, if I'll be able to connect. But if not, as you like record, I'll just go back and look at the recording. But Thursday, I should definitely make it because we'll be in the hotel and everything else.
Enjoy your trip.
Oh, yeah, it's a quick one, but it's a lot of packing and a lot of getting together stuff and trying to make sure school is straight before I go.
Enjoy the warm weather.
Yeah, it's supposed to be 70s, so
Wow.
I'm excited because I'm
my
not,
sister lives in Las Vegas. Enjoy it.
yeah, I'm not a cold person. So, but yeah, just if I'm absent, that's
Okay,
why.
I understand.
Thank you.
Okay, be safe.
Professor, please, I have a question.
Go
I
ahead.
already registered for anatomy 122 for
Yeah.
the Ruth Negley and Nancy Konoka.
Okay.
So you say you will confirm.
I will confirm it. I thought it was the Friday class, but Toyin, if you're saying that it's the Monday, Wednesday class, it probably is. And
No,
I just
it's
need to-
not. It's only Thursday for the practical. The lecture is virtual.
Well, that's the class with Ruth Negley. Mine will not be virtual. Mine
You
will
said
not
what?
be virtual.
Oh,
Mine,
do you?
mine will
okay,
not be virtual like that. It will be online, but they'll both be scheduled classes.
Do you mean you want to take 122?
I am teaching 122, but I
Oh,
can't
okay.
remember which section I'm teaching. I'll have to ask. I thought it was the Friday one, but I don't know for sure.
Okay, so next class you confirm it for us,
I
right?
will confirm it. I'll actually send you an email too.
All right, thank you.
You're welcome. We'll see you later. Kevin, Carrington, or Shayla, are you here?